So good morning, everyone. As Lang said, I am Ravina. I'm a final year clinical trials apprentice at Syneos Health. So today I'll be presenting a brief overview of my dissertation titled An Analysis of Recruitment Strategies Used in a Pediatric COVID-19 Vaccine Study. So why have I chosen to perform analysis of recruitment strategies used in a pediatric COVID-19 vaccine? So first and foremost, it is vital to understand the relevance and importance of recruitment in developing novel treatments. So drug development timelines are quite consuming and costly, and they can cost anywhere up to $2 billion US and take around up to 10 to 15 years. All new drugs, treatments, and vaccines must undergo testing in human participants through clinical trials, and there are various processes as seen on the slide. So from startup to implementation to closeout. So recruitment of participants in the trial is done at the implementation stage, after the trial has been approved by regulatory bodies and clinical trial sites, such as hospitals, have been activated to run the trial. It is important to note that recruitment must be considered at the start of a trial at the study design stage, as recruiting a low number of participants or the wrong participants will disrupt the integrity of the trial. So what are the challenges of recruiting participants in clinical trials? Despite recruitment being a driving factor, recruitment of participants often accounts for a significant portion of drug development and can take up to 30% of development timelines and cost up to $1.2 billion. This is because most clinical trials experience delays of 1 to 6 months due to difficulty recruiting participants, with 11% of clinical trial sites failing to enroll a single participant and 37% under-enrolling. Recruitment is therefore arguably one of the most important aspects of bringing a new therapy to market. The aim of this project was to provide an analysis of the recruitment strategies used in a pediatric COVID-19 vaccine study to optimize recruitment rates and ultimately reduce study timelines. In order to appreciate the context of this analysis for this specific experience, there is a need to understand why a novel COVID-19 vaccine was required. So in late 2019, the most widespread coronavirus was discovered in Wuhan, China, hence termed COVID-19. Spread through human-to-human transmission caused by the virus severe acute respiratory syndrome coronavirus 2. So by March 2020, COVID-19 was declared a global pandemic by the WHO. Due to the significant economic and health burdens as a result of the pandemic, several vaccines were deployed and gained emergency use authorization in late 2020 and 2021. Emergency use authorization implies that approval for new drugs and vaccines still under development were issued in a public health emergency. Since COVID-19 pandemic started, over 773 million cases, 1 million hospitalizations, and 7 million deaths have been reported globally to date, with 13.59 billion vaccines doses administered. However, even with the high vaccination rate, there is still a large global need for additional vaccine doses to protect against evolving variants of COVID-19, as well as vaccines with better storage conditions. Also, it is not known how long current vaccines provide immunity against COVID-19, resulting in the need for booster vaccines, and there is also a need for suitable vaccines for sensitive populations, such as in children. Therefore, this project will be focused on pediatrics. So what is the COVID-19 vaccine I'll be looking into? So the vaccine being developed is a protein-based vaccine targeting SARS-CoV-2, the virus that causes COVID-19. Recombinant technology is used to create vaccine nanoparticles, which is then mixed with matrix M adjuvant to enhance immunogenicity to create the ready-to-use COVID-19 vaccine. Subunit vaccines include only part of the virus that best stimulate the immune system. This type of COVID-19 vaccine contains harmless spike proteins. Once the immune system recognizes spike proteins, it creates antibodies and defensive white blood cells. If one becomes infected with the COVID-19 virus, the antibodies will fight the virus. So at the moment, there are only 2 COVID-19 vaccines licensed in most countries and recommended by the World Health Organization for pediatric population, and they both use mRNA technology. So that's Moderna COVID-19 vaccine and Pfizer BioNTech COVID-19 vaccine. So mRNA vaccine technology is new and long-term adverse effects in humans are still unknown. Due to the rapid development, there is a negative public perception. Despite showing 95% efficacy in both vaccines, it has not been historically widely used as a vaccine due to liability of RNA, which is typically degraded by ribonuclease. Now I've explained the background of the vaccine, it is important to understand the perception of participating in clinical trials in determining recruitment strategies. Healthy participants tend to be more challenging to recruit due to concerns around risks associated with the study drug. So this means they do not have the immediate benefit of improved health versus risk as healthy participants. So for example, a phase 3 oncology trial. From viewing literature, the most common recruitment barrier found was study protocol complexity and lack of awareness of clinical trials. The reasons for participating included improved medical care, contributing to scientific research, and trust in study doctor, as well as financial gain. However, with financial gain, there is a risk of financial coercion, as the finances provided to participants are typically given to cover inconvenience and time spent on the study. This is overseen by ethics committee to ensure no financial benefit is given, only in reimbursement for participation, as stated in ICH-GCP. So due to the lack of papers available for recruiting pediatrics to a vaccine clinical trial and the findings from the previous slides, I concluded the following aims. So my primary aim was to analyze the weight of the following in recruitment, so protocol and study understanding, healthcare providers, clinical trial sites relationship, and digital outreach. And the secondary aim was to reflect on recruitment practices that were both successful and inefficient for this pediatric COVID-19 vaccine. The overall purpose of this project was to address stakeholders interested and influenced by the research to optimize recruitment. So first, it's important to understand the study design itself for the study. So this project is based on a phase 2/3 randomized observer-blind placebo-controlled age de-escalation trial to evaluate the safety and immunogenicity of 2 primary doses and a booster dose of the COVID-19 vaccine, given 21 days apart in pediatric populations. So what does that all mean? So participants were randomly assigned to receive either the COVID-19 vaccine or placebo during the initial vaccination period. So the first injection was given during the first 2 visits on the site, so 21 days apart. So participants, their parents and guardians, and the study doctor and staff did not know about the vaccines the participants were receiving at the time. However, they found out 6 months later. About 6 months after enrollment in the study, children who initially received placebo will receive additional 2 doses of the study vaccine. Those who initially received study vaccine either received placebo and then about a month later another dose booster of vaccine, or received a booster dose of study vaccine. So all participants eventually received 2 primary doses and a booster dose of study vaccine. So this was the following timeline that was planned. So it displays the planned study timeline before the recruitment campaign. So FPI being the first patient in, marking the start of the recruitment campaign, and LPI being the last patient in, marking the end of the recruitment campaign. So the aim was to recruit a total of 3,600 participants. And the countries anticipated were Colombia, Indonesia, Mexico, Peru, South Africa, Spain, and the United States. And these were determined via a feasibility testing. So considering competitive landscape, site startup timelines, vaccination metrics, regulatory intelligence, and disease metrics. So what was the pediatric engagement strategy that we used? So in order to assess the primary aim, the following strategy was used in the study. Protocol and study design understanding using patient-facing materials. These materials are designed to support discussions with families and study participants, answering questions before they are asked. Healthcare providers, clinical trial site relationship through site-facing materials and site-specific recruitment plans. So these plans are used to understand where the site requires support and digital outreach via use of vendors. So utilization of social media campaign and utilization of search engine optimization campaign to optimize recruitment for the study based on the feedback from the site-specific recruitment plans. So the following patient materials were used. So again, supporting patient, family, and education. So this is just an example of one patient brochures we used, and mention of patient advocacy groups. So for example, advocacy groups such as Connect for Children in the EU could be used. So the materials were designed by for patient understanding of the study. So patient brochure for awareness, and then flyers. Materials are also designed for site staff to assist in easy access to information. So, for example, doctor-to-doctor letters or schedule event cards. So the site-specific recruitment plan is a questionnaire given to sites prior to the site initiation visit to assess this type of support they require. So questions could include things such as who's responsible for patient recruitment, what are the anticipated challenges, are they interested in central media campaign, and have they ever used recruitment materials prior? So just getting an idea on where the site is with the child and if they have previous experience. So the findings we had from the site-specific recruitment plans was the study procedure, so the requirement of 10 milliliters of blood sample. History of COVID-19 and placebo concerns were also highlighted. The inclusion and exclusion criteria, so young population, especially in Honduras and US, where there are competitors, and parent hesitation, so misinformation. From a recruitment perspective, these challenges are important to consider whilst designing a campaign. However, not all challenges can be addressed due to gathering statistically sound data. So, for example, change of protocol unless deemed a safety issue by regulatory agencies. In terms of central media campaigns, US and Mexico expressed their interest. However, as Mexico was used during the end of the study, it was not possible from a feasibility perspective. However, the US did adopt a digital outreach campaign. So 2 digital outreach campaigns were used. So initially we used a digital media campaign. So this was placing advertisements on Instagram, Facebook. So patients would click the link and they'll be referred to a landing page where they can see all the study details and where they can provide their own details if interested. They are then contacted by a specialist within 24 hours and then referred to sites if they pass the screener. Search engine optimization uses a different approach. So it uses online browser behavior and geotargets to identify interested participants. So this tends to be a banner advertisement, which patients can click on. And again, they will be referred to a landing page where they provide their details and complete an online screener. And qualified patients are referred to sites by email and portal. So for the digital outreach campaign, which was exclusively used in the US, due to the interest from the SSRPs, the social media approach experienced low levels of screening and randomizations despite pre-screenings having a steady increase. On average, there was 1 patient randomized a week despite increased referrals. So due to the poor recruitment numbers, this approach was dropped. We then adopted a search engine optimization campaign, which was used throughout the entire study, and it gained over 13,000 referrals, which is really impressive. So overall, this was a successful recruitment campaign despite a few bumps, with a total of 3,752 participants recruited, exceeding the recruitment estimates of 3,600 by 4% despite shifting countries as highlighted in slide 13, so back where there was the site-specific feedback. The last patient in date was extended due to a number of vaccines available at site and site activation rate, but this was not due to the recruitment campaign, as the target participants were already met. So overall, the study was a huge success and determined protocol and study understanding, clinical trial site relationship and digital outreach were equally important and go hand in hand, as well as the recruitment practices were successful for this pediatric COVID-19 vaccine study and should be implemented in future studies. By adopting this strategy that suits the indication best, so for example, healthy participants receiving a COVID-19 vaccine search engine optimization campaign was more successful than the digital media campaign, as well as working closely with stakeholders throughout the campaign, such as working closely with sites via the site-specific recruitment plan, leading to a successful campaign by identifying what strategies are required. Overall, recruitment campaigns should be considered from study design in order to implement patient-first attitude, identify what patients require in addition to gain scientifically sound data. Study teams must also be highly adaptable. So as seen in this study strategies, they did shift throughout. So for example, dropping the digital media approach. So overall, pharmaceutical companies, sites, and others involved in clinical trials benefit from this by incorporating recruitment strategies discussed to save money and achieve or shorten study timelines, in addition to enhancing patient and communities' trust and understanding of clinical trials with patient-first attitude implemented. So I'd like to thank the following people for all their help. So Lynn, my academic supervisor, Jenny, my line manager, my mentor, Sean and Rebecca, the entire apprenticeship team, Syneos Health and the University of Kent. Thank you so much.
Amazing, Ravina. Thank you so much for this, for your amazing talk, for your project, and for your efforts to present today for everyone your exciting project. And do we have any questions to Ravina? I think that I've seen something already. Maybe I'm mistaken the chat. Please, if you don't want to use the chat, just raise your hand or just start talking, asking questions to Ravina. Anybody would like any specific questions about the project? If not, maybe I will just sort of go with the first. Oh, Kirsty, sorry. I think the Kirsty looks like. Oh, sorry, we've got one as well.
Yeah, I was just going to, but I think Christine's just come with a question.
So Christine is asking, what are the regulations regarding informed consent, and are children given an option whether to opt in or not? It's a good question.
Really good question. So we do have assent forms. So because children technically can't give consent via the normal consent form, they tend to be like 12 pages plus. We have one with little images, really easy to understand. It shows images of what will happen throughout the study, and if they understand that there will be a blood taken, there will be an injection, they might catch a cold, and all the things that may be covered, and they do sign that off. Again, it depends on the age group. If they're under 1 year old, they can't really do that. But if they are able to understand it, they do sign that off. But in terms of regulations, it depends by country. It really depends globally. But thank you. Really good question.
Any more questions? What was the most challenging part of your project, Ravina?
I think the most challenging part was just getting the right strategy. It's just the going back and forth, especially with the US. It was really, really tough recruiting in the US just because all the stigma associated with COVID-19 and clinical trials. So trying to convince parents that this is not going to be harmful. We've tested before. It's a phase 2-3 study. They're in safe hands. So just trying to build that trust with people.
Yes. We were talking about this week with our MSc students about the role of the sharing the data that's got on the drug discovery and development and approval of new medicines. We're talking about also the drugs, NO1 drugs, when they are developed for specific patients. And for those patients, it can happen within 10 months, recent drug for the personalized medicine. So it's quite different, isn't it? Whereas there's a lot of regulatory sort of obstacles on the way to approve the drugs, but also like when you look at the regulations and when you look at clinical trials to get the data, isn't it, to have the consent and to be able to analyze is another obstacle. So I think that's quite heartbreaking in a way. That it's all about sharing, isn't it?
Yeah, definitely.
Perfect. Sorry, say again.
I was going to say the advocacy work that we're doing is really good because it is sharing data with charities and it's sharing data with people that represent the community. So of course we were doing healthy volunteers, but for other clinical trials that have a subset population that are unhealthy, it's really representing everyone and increasing diversity as well.
Perfect. Another question, do you see a lot of development of artificial intelligence technologies in clinical trials in terms of to speed up the process of analysis and looking into that as well in Syneos Health?
I'm not sure what I can say, but I've seen some in the pipeline. So
yes, we've been talking about this. Actually, it's a move forward, isn't it, with every drug discovery process? So yeah, it's interesting. Right. Any more questions? Have we got spare 4 minutes?
There's some question. There's a question, or well, 2 questions I think from Casey in the chat.
Perfect. Thank you, Kirsty. I didn't see that. So Casey is asking, would the type of recruitment campaign used depend on the type of demographics you are trying to recruit? Or is one type of strategy always going to be better? I guess as well, would stuff like fake news have an impact in clinical trial recruitment, trust, and therapeutic options?
So yeah, demographic definitely depends on what strategy we use. So that's why we implement the site-specific recruitment plans. So you get an understanding from the site what they need. So for example, an Alzheimer's study, maybe digital media would be better because it would be something that pop up on Instagram. You might be like, oh, my mom has Alzheimer's or my dad has Alzheimer's. So it really reaches the right target audience. Whereas for a COVID-19 vaccine for pediatrics, that's not something people actively would be interested in clicking on. If they've scrolled through Instagram, that would be probably something they scroll past. Whereas if it's coming to people that are actively searching for COVID-19, it may be better. So it really depends, different demographics, even the ages, that's pediatric. Maybe if it was an older population, a different strategy would work. But usually we do use the site-specific recruitment plans, getting it from the experts, which are the sites that deal with these patients on a daily, and they give us input. And then the question about the fake news, I guess it does, because at the end of the day, participants are just everyday people, just like you and I. Anyone here can participate in a clinical trial. So depending on how you feel about clinical trials will sway if you want to participate, or if you're on the fence, or if you're really eager to participate. So all of it is involved.
Definitely.
Very good question. Thank you.
Perfect. So I guess if there's no any further question, unless something's
Nothing.
happened. Oh, there's one hand up.
Yeah.
Oh, it's not much, it's not a question. I just have a comment regarding the AI technology. So, in terms of advancement in technology, so AI can be used to kind of predict the new variants of a particular pathogen. So we have predictive modelings in place. So you know how they have different variants of the COVID strain. So you have the alpha, beta, gamma. So we have AI to detect those variants now, and that kind of helps in the vaccine development process for COVID.
That's
Amazing.
amazing.
Thank you for that. Yes. There'll be something that it moves forward anyways through the CTS, for clinical trials. Yeah,
Absolutely.
definitely.